Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about the reproduction of Porifera. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So basically, sponges which are included in the phylum Porifera, they reproduce both asexually and sexually. Asexual reproduction in sponges is by 5 different ways: regeneration, formation of reduction bodies, budding, fission, and formation of gametes. Let's talk about them one by one. First one is regeneration. So we all know what is regeneration. The power of regeneration is very great in sponges. Any cut part will regenerate the whole sponge. So if you fragment sponge in different parts, each part has the capability to generate the new body, that is called regeneration. Next is formation of reduction bodies. So many sponges disintegrate in adverse circumstances. That means they are getting destroyed in harsh temperatures or harsh conditions. This is called disintegration. The disintegrating sponge will usually collapse, leaving small rounded balls called reduction bodies. So after the disintegration, you will find few such bodies which are called reduction bodies. And reduction bodies mainly contain two parts. The outside part is called pinacoderm, and inside there are few cells which are called amoebocytes, which are basically totipotent cells. Totipotent cells means these are stem cells. So we all know that stem cells have the capability to produce the new cells and ultimately to produce the new animal. So each reduction body consists of an internal mass of amoebocytes. So these are amoebocyte cells which are covered externally by a pinacoderm. When the favorable conditions return, these reduction bodies grow into complete new sponges, and that is possible due to these totipotent cells, due to these amoebocytes. So they will form the new sponge bodies in the favorable condition. Next type is called budding. So numerous archaeocytes gather near the surface, resulting in small outgrowth of the pinacoderm. So pinacoderm is basically the outer wall of the sponge, and archaeocytes, which are also known as amoebocytes, and just now I have mentioned that amoebocytes are nothing but few totipotent cells. So sometimes these amoebocytes or archaeocytes gather near the surface just like this. So they gather near the surface and that results in the small outgrowth. So here is the small outgrowth you can see in this picture, and this is called bud. And this bud, the sperm grows outward to produce a small individual which gets detached to form a new sponge by fixing itself to a suitable substratum. So these archaeocytes or amoebocytes, they will gather in these bud and they will form a small bud, and that bud will be grown up and that will form this thing. This is the outgrowth, and that will be detached from the main animal body, and here is the new sponge which will fix it and it will become the new animal. Next type is fission. So parts of sponge body are thrown off from the sponge body. Just like this part is thrown off from the sponge body, and the part of the parental sponge thus thrown off develops into an adult individual by fixing itself to a suitable substratum. So once this part is broken down, this part is getting thrown off. It will be fixed like this with the substratum and it will form the new sponge body. Next is formation of gametes. So gametes are internal buds formed within the sponge body. So in this case, in budding, we have seen that this bud is formed, that is external bud, right? But gametes are basically internal buds that are formed within the sponge body. And they get detached when the parent sponge is decayed. So once the parent sponge is disintegrating, it is decaying, that time that gamule is getting detached from the parent body. And this gamule is basically a small, round, hard ball, just like this, consisting of internal mass of archaeocytes. Again, here are the archaeocytes because these are the main stem cells. So these are important. These archaeocytes are surrounded by double membrane. So here is the outer membrane and here is the inner membrane. So these archaeocytes are surrounded by double membrane, outer as well as inner membrane. And it has a small outlet. This ganule has a small outlet. You can see here. This is called micropyle. Okay. Now what happens basically in autumn? Sponges suffering from cold and food scarcity, and they get disintegrated, leaving behind number of granules which remain inert throughout the winter. So these granules are just like bacterial or fungal spores. They stay inert throughout the harsh condition throughout the winter, and when is the food scarcity, they will remain as it is. Now in spring, when the conditions become suitable, the granules begin to hatch and living contents of the granules escape out through the micropyle and form the new sponge. So when there is the favorable condition, what happens? These archaeocytes, which are the main stem cells which are staying inside the granule, they will come out through this micropyle, and since they are stem cells, they will form the main animal body. So these are five different types of asexual reproduction in sponge. Now I will talk about the sexual reproduction in sponge. So sexual reproduction involves formation of sperms and ova. Since it is sexual reproduction, it involves gametes, right? So sperms and ova are formed in sponge body, and sponges are mainly hermaphrodite. Hermaphrodite means they are bisexual. Bisexual, that means both eggs and sperms are produced in the same body. So you can think that self fertilization will occur, but no. There is the cross fertilization. Although they are hermaphrodite, but always cross fertilization will occur. Why? Because eggs and sperms are produced at different times. So, for example, if eggs are produced in January, the sperms will produce in March. This is just an example, right? So please don't take it seriously. It's just an example. So for that, the self-fertilization is not possible. Always cross-fertilization will occur. So eggs are produced inside the body and remain inside, waiting for fertilization. So basically, eggs will not leave the sponge body. They will not move. Whereas sperms are produced inside the body and they leave the body through osculum. So you know that if this is the sponge body, this is the big hole that is called osculum. So suppose sperms are produced here, so they will come out through the osculum. And this is body, sponge body 1, and suppose this is sponge body 2. Now what happens? These sperms will come out through the osculum and they will swim in the water and they will then enter the body of another sponge. Like in this case, this is sponge number 2, where these sperms will enter, and they will enter through water canal system that I am not explaining over here, and reach the eggs to fuse them, ensuring internal fertilization. So in sponge 2, there is the egg that is waiting for the fertilization. So sperms will come from sponge 1 and they will enter the body of sponge 2, and they will fertilize the egg. And in this way, the fertilization occurs. So this is absolutely the cross fertilization, not the self fertilization. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.